So thank you for inviting me and for organizing this. I will, as it was said, I will talk about a different perspective in fabrication, one that is growth-based. Yes, so I was introduced. I'm currently doing my postdoctorate research at Edeka after finishing my PhD at Lund University in biodigital matter. So we've all know this and heard it before, but the current data that we have on the climate crisis brought a lot of attention to the construction industry and the way we're related to the carbon cycling. And besides a lot of different strategies that are needed and applied right now, I think it's really important to reconsider our material culture. And this is because in 2020, the ratio of the anthropogenic mass as it relates to the entire biomass on the planet has already surpassed it. And this is, I think, because it's really hard to conceptualize and to grasp, considering the immensity of the Earth and then how we perceive the infinite natural resources that we have. So in the 1900s, the entire of what we built, like concrete and metals and asphalt and all the processed material on Earth, was 3% of the biomass. And in 2020, it was already higher than it. And in 15 years, it's going to be 3 times as big. So this is just to understand a bit how we're relating and what is our impact as a construction industry to it. Because with this biomass and the role of biology in mind, we can see that since the beginning of humanity, we have been influenced by it and we have been fascinated by it. And even in our early dwellings, in the caves, we've been painting animals, and then throughout in the ancient history, motifs of animals and plants have carried through the acanthus leaf and the Corinthian column, then the Art Nouveau, vegetation, and Baroque. Like it's up to the current times where we move from biomorphism, which is just copying in 2D or 3D vegetation or animals' biological system, to getting inspired from the function. So in biomimetics, these are some examples of biomimetics. We're looking at taking functions from biological structures and applying them into building systems. And while this is interesting and it is a step further, it is far from the potential that we can achieve by really understanding and taking on what biological and natural systems can do into our built environment. And while we've seen examples of large scale, so implementing biological systems at large scale, I think there's a lot of potential in the material systems as well, and especially at all scales. So how do we work with multiple scales, and how do you address these issues at the same time? So this is a concept of transcalarity, which is what I developed during my PhD, which is an organizational principle in complex systems that observes interdependent behaviors between scales. But it is also a design strategy that targets several scales simultaneously while considering the interdependence. So the journey of my PhD started in Namibia, where together with David Andreen, we went to study termite mounds. And this is actually because they're a really amazing example of transcalarity and how behaviors at lots of different scales stack up and create a highly exquisite superorganism that has really impressive function. So we studied the termite mound physiology in 2 ways, looking at the geometry and function, and then at the materiality. Of course, ideally, these are interconnected, but they just had different focus in 2 different projects. So the first one is meristem wall. And here we 3D printed a large, at full scale, a building facade. And this is inspired by the termite mound wall, because it is sometimes believed that the termite mounds act as a thermal, that they modified thermal environment inside versus the outside. That is not actually the case, but they act as if they are the lungs of a large organism. So to breathe the metabolism of all the termites and that live inside. So in this way, we've also created so instead of being a wall that separates the inside from the outside, it's actually a network of interconnected channels. And these can be actuated with very low volume, with very low energy, to move air from the inside to the outside. And it's also integrating different functions inside, such as piping and electricity. So in the typical AM multifunctional, and also opens up to the outside, where it can be like a bioreceptive wall for urban biodiversity. And yeah, we also collaborated with Mariana Popescu for the interior CNC membrane that acts as a filter for the channel openings. And this project was exhibited at Venice Biennale. But another way that we looked at this, the functionality and the way we've got inspiration from the termite mounds, is the material of it. Because so the termites themselves, they don't have a digestive system that can break down cellulose. So what they do is instead they've made a symbiosis with a species of fungus called Territomyces, which lives inside the mound. So what happens is that the termites harvest plant matter and bring it inside the termite mound. And here there's a species of fungus that slowly breaks down the material, so that both the termites and the fungus can digest and live off of it. And this is why I'm talking about this, is because it's quite an interesting material. It's quite lightweight. It has it's hydrophobic and pretty strong considering its weight. So we started here to see if we can replicate it and what can we learn from this system. And it is interesting because it is built additively. So the termites add it one piece at a time, and how this object in the end, this structure, ends up negotiating a lot of different needs. So the need of the fungus to breathe, so to have access to oxygen, the needs of the termites to access it for deposition and removal, and of course the aggregation of the plant matter. So then what we did, we went to Namibia, as I said, and we opened a termite mound and took samples, lots of samples from inside of it, and then smuggled these samples in a toiletry bag and brought it home in the lab, and then we isolated. And we also managed to get the original fungus that lived inside there. And then we've added the substrate for this, which is plant fibers. In this case, it was cellulose and wood chips, but lots of different substrates are viable. And then we 3D printed with it. So at this stage, this is essentially a wood-based 3D printing, which is in our case, it was a very soft, wet material that on its own didn't really have much stability. But after the 3D printing, there goes a growth stage. And this is where you can see in the middle, the fungus grows on the surface, and through the mycelium, which is the root network of it, essentially binds the material together. And what is perhaps even more interesting is that it not only acts as essentially a reinforcement of the wood particles, but it's also changing it at a chemical level. So you can see from the control, our main species that we ended up working with, even after being submerged for 24 hours in water, it doesn't decompose. It maintains its mechanical integrity because it is not just wood pulp anymore. And so we took this material system and put it together in a full-scale 2-meter column to scale up the process. So this project we started in 2017. So it's quite some time ago that we've been trying to address these issues. So I think what's important to get here is that how working with a living organism kind of really forced us to try to address the different scales, because you cannot work at one and then expect it to just solve itself across the different scales. We needed to negotiate the needs of the fungus, which meant increasing the surface area, because we wanted to maximize the enzymes that were secreted by the fungus. Because the more enzymes are secreted, then the higher the transformation rate of the material. And this is why, so understanding what happens at the very small scale, molecular level, down to negotiating. So here you can see how we maximize the surface area so that the fungus has access to oxygen to breathe during its growth stage. And also this helps for desiccation, so to ensure more even drying with no crackings or less deformations. And these are some of the parameters that we worked with. So these are spanning the printing parameters, extrusion, to the material composition, to have it both being extrudable, but also biocompatible with the fungus. And then, of course, the design parameters. So all had to be negotiated somehow simultaneously. So to conclude, it is clear that we need, of course, lots of different strategies for addressing the current state, but the material rethinking and how the material integrates with the design, I think it's really relevant. Yes, and I think, of course, it's not easy to work through all the scales simultaneously. But I think that now we've reached a point where we can do this, both because of the computational power that we have access to and to the tools of fabrication that can materialize these intricate interdependencies that we have. And also, I think it's really important to work across disciplines, because only like this can we bridge the different needs from like very small material scale to global scales of material flows and information. So yes, and this is a project that was already completed. So right now, I've started my postdoc at Kassel just last month. So now we're in the process of scaling up these biomaterials, so testing different materials, testing different species of fungi and bacteria, and then also we're building a lab that has both the capacity for microbiology and for architectural towards large-scale prototyping. So I think because these interfaces are really important to be able to create these kinds of projects. So yeah, this is where we're at. So stay tuned for the next project. Thank you.
So hands up.
Thank you. Again, a very interesting approach. I'm curious, how do you envision the longer term usage of these parts? does the mycelium continue to grow? Is it sterilized at some point? How do the material properties evolve over time as the ratio between mycelium and wood changes and things like that? Lots of open questions there for me.
Yes, these are very good questions. I don't think there is right now the one answer for it. What we've done in this case is stop the grace by drying and killing the mycelium, which, because even as it is, bringing mycelium into housing raises some issues. So at least if you can say it's inert, this is one way to keep developing and not have too much pushback from users and the industry. But I think there is perhaps, there are ways in which the fungus can be kept maybe inactive, but still alive. And I think this depends a lot of the conditions of growth and the fungal species. So there's a lot of parameters that need to be discovered and put together. But in this case, it would really help. It would bring even further benefits, such as the capacity to self-repair, right? Like we have examples of concrete with bacteria that when it cracks, it can be regrown. So I think by working, but of course, it requires a lot of setup for the microorganism to be able to, that we control it in a way that it doesn't keep breaking it down. But of course, there's also the issues of longevity. And of course, this will not last as concrete. So this means that there is a more need of, because it is the potential is that it uses waste and it is very low embodied material requirements. And it is, we're working on making it pretty more easier and less CO2 for growing. But then of course it doesn't last as long. So I think another way of looking at an approach is not necessarily making everything last forever, but if it's less cost to make, then we can replace it more often. And then this also comes to issues of care and repair and maintenance around buildings. Like, there's a lot of things there. But I want to share.
All right. Anyone else? Oh, just at the farthest point of the room. You can use your own mic.
So I have a quick question about the thickness. So what we saw was a pretty thin layer which is printed. So how scalable is it? How thick can the layers be? And will it have an influence on the drying process and the growth?
Yes, it's a very good question. Well, we started with a thickness that actually came close to the original biological model, which is the fungal cone, because we thought, so the goal was to maximize the amount of fungal growth and the biotransformation. And it was somewhere around 5 millimeters that the growth was maximized on the inside as well. Because in the middle of it, there's almost no oxygen. So therefore, there's very minimal growth. But then it can be transported if the thickness is not so high. So this is how we started. But of course, in terms of speed and throughput that it can be done, of course, it's highly limited. But we have, on the other hand, right now, the mycelium biocomposites are deployed at quite a large scale through casting. So this is a more common way to produce them, is just to make molds and then cast them. Even in large volumes, they're currently used for like packaging and insulation and things like that. So I think there is a balance to be had between the speed of it and the maximizing the growth and the transformation. But of course, it depends on the use case too. So I think this is why it's actually the possibility for customizing the material properties for the use and for the part that is really high. And of course, there's lots of other different strategies. So we're exploring different ways of 3D printing. So going into binder jetting with it, which kind of has different set of requirements and processes. But I hope this answered.
All right. One question raises more questions. Hi, thank you very much. One question idea. I assume you basically have a pulp and you mix the mycelium into the pulp and then extrude it afterwards. Could you also imagine to make the mycelium selective, so to say, that you have different areas which have different mycelium content with different growth rates, so to say, and then also the material changing over time, or some stuff washing out, for instance, or stuff like that?
Yes, these are very good points. Actually, this is part of what we're planning to do in the next stage, because I think by having a living agent inside the material, I think we can unlock, I believe, the next level of additive manufacturing, which is actually customizing within. So not just through geometry, but by changing the material properties. And I think there's several ways, by changing the ratio of nutrients, by guiding it, potentially even having stronger. So these are all really potential and really exciting options. But yeah, we're not there yet.
Okay, that was your hand raised?
Thank you. As far as I understood, you took the mycelium that you found originally from the termites and used it here. And I imagine that you did not find only one stem of mycelium, but different mycelia. And did you also look, or are you planning to look into a directed evolution where you have an idea of which mycelium you would like to use or which would be better to use? Or do you see?
Yes.
Both there?
Yeah, another very good question. So we started because the properties of the material of the fungus comb itself, we had samples, and this is really fascinating. And so we tried to replicate that. So this is how the project started. We isolated the fungus, and all the tests were with that. But unfortunately, that didn't end up leading much further, because as much as we could grow it in laboratory conditions, when moving it to larger scale, it didn't really work. And I think in retrospective, it does make sense that it's a 40 million years evolution of this symbiosis between the termites and the fungus. So trying to grow a partner that has evolved without the other one didn't really work. And also there's mycologists that are working on doing this for the purpose of growing the mushrooms, because they're actually really good. Apparently they taste really well, and they're quite large. So just from the food perspective, it would be good. And that hasn't been managed yet. So we took it to a step, and then we had to open up the search. So we've done a few other different species that we took in parallel, and then the ones that had the highest speed of growth and the most resistance to contaminants. Because this was also the problem. As much as it would grow, it's a species that has evolved to grow very slowly, so that the termites also have time to eat from the sugars that it breaks down. But then in this case, we don't want in fabrication a fungus that grows very slow. So we spread it out, and we tried a white rod, a brown rod, and a litter decomposer as different strategies to see a bit of the breadth of it. But of course, there's an estimate that there's around, a conservative estimate is that there are 5 million species of fungi in the world. And a lot of the tests that have been done in biomaterials are covering maximum a dozen. So I think there's an incredible opportunity to go through this, because they have such a high different behavior, growth rate, different enzymes that secrete. So I think there's an incredible potential to be discovered still in this field. And yes, the fungal species is actually one of the key aspects.
Okay, thank you very much, Anna.
That's good.